In this video lecture, we're going to talk about DNA replication. And in order to talk about DNA replication, we have to also talk about the chromosomes themselves and how that works. So basically, we compare the prokaryotic cell with the eukaryotic cell. There's quite a bit of difference. Obviously, the prokaryotic cell is very simple. It does not have a true nucleus, so the DNA material has to be located someplace other than a nucleus. And what we call the prokaryotic cell, I kind of called it the soup cell because everything's kind of sloshed around in the cytoplasm without membranes around them. So the DNA is no different. The DNA is located in the cytoplasm, and the DNA is basically like a circle. It's a loop, and it contains all the genetic information that the bacterium needs to survive, but it's not in long chains like we know as chromosomes. It's in a single loop. And as you can see over here, this single loop, this is called a plasmid. They don't give you the name here, but it's called a plasmid, and it's basically the same as the DNA in a eukaryotic cell in that it base pairs the same, cytosine with guanine, thymine with adenine. But in eukaryotic cells, you have obviously a lot more DNA because of the complexity. As the book says, 1,000 times more DNA than prokaryotes has, because there's just a ton of extra things to deal with in a eukaryotic cell as opposed to a prokaryotic. Eukaryotic DNA is located in the cell's nucleus. It's going to be basically located on the chromosomes themselves. Now, the number of chromosomes varies widely from one species to the next. We've already talked about the number of chromosomes in a fruit fly, the Drosophila, when we talked about DNA in the first chapter with Mendel, and we said they had 8 chromosomes. We, human beings, are going to have 46 chromosomes. So every organism is going to have their set number of chromosomes, and usually larger the organism, the more information has to be stored, so the more chromosomes there would be. In eukaryotic cells, well, like we've already talked about, whenever the DNA is in a non-dividing cell, it's basically in an uncoiled position. Basically, you can't tell one chromosome from another because it's like the spool of thread that's unwound, all stuck together in a big ball. That's called chromatin. But when the DNA is going to separate, when it's going to replicate and then separate in the process of cell division, it then starts coiling on top of itself and becomes what we call chromosomes. Now, the chromosomes are going to consist of the DNA, but it's going to be tightly coiled around proteins that are called histones. I kind of look at histones like they're hangers. They're kind of like a hanger that you hang your DNA on. The DNA and the histone molecules form nucleosomes. That's not a term you're going to see widely used. This is the first time I've seen it used is in this book, but generally speaking, that's what it's called. When the nucleosomes pack together are going to form a thick fiber, and you can see here these thick fibers, these coils, are going to turn into supercoils because the coiling of the DNA around the histones, the hangers, is going to then coil again and into a histone, into a nucleosome. The nucleosomes coil again on top of supercoils, and the supercoils coil to make a very thick fiber called a chromosome. So as you can see, from a very thin fiber of DNA, it coils and coils and coils on top of itself until eventually it becomes very, very thick and noticeable and stainable and noticeable under the microscope, and we call these chromosomes. So during DNA replication, and by the way, each cell has to make a new set of DNA every time the cell wants to divide. So the double helix of the DNA is going to have all the information that's needed to reconstruct a new strand of DNA. One of the strands of DNA of the double helix is what we're going to be calling the template, and the other side would be referred to as the dummy DNA. I'm not going to get into a long explanation of that, but the one side is the true template for the message. And when we get into protein synthesis, we'll talk more about that. If we're in a prokaryotic cell, the DNA replication is going to begin at a single point. It's going to continue in two directions until it's done. But in a eukaryotic cell, the DNA is going to occur at lots and lots of places along the chromosomes at the same time. And this replication is going to proceed in both directions until the chromosome is completely copied. The sites where the separation and replication occur are called replication forks. And that's basically because they separate. You have a fork like a fork in the road. They separate the two strands of the helix, and then each new strand gets a new strand by base pairing. So before each cell is going to divide, it's got to make an exact copy of DNA so that each new cell is going to have the exact number of chromosomes that it's going to need to survive. So it has to have a complete set of DNA. Otherwise, every cell division would have the number of chromosomes, and eventually you wouldn't have much. So during DNA replication, the DNA molecule is going to do a number of things. First of all, it's going to unzip itself down the middle. It's going to separate into two strands at each point where it's going to replicate, and then it's going to produce two complementary strands following the rules of base pairing, A with T and C with G. Each strand of the double helix of DNA serves as a template for the new strand. So they're going to separate, and then each new strand will fill in its complementary bases until it has an exact copy of the DNA. So as you can see here, you have a replication fork where it has unzipped itself. New nucleotides are popping in at their appropriate places all along here, and it'll work in both directions. So you have a replication fork on each end of this little opening where the actual replication occurs is called a chromosome puff. So if you looked at microscopically, if you looked at a chromosome that was replicating, you'd see at various intervals along the chromosome, you'd see this little, and it's called a puff because it looks like a puff, like a little explosion went on there. And that's because it's unzipped itself and it's in the process of plugging in new nucleotides on each side of the DNA. So first of all, the DNA replication starts out by unzipping itself down the middle, and it unzips itself by breaking the hydrogen bonds between the base pairs. So A and T and C and G are going to have hydrogen bonds between them, and to start this process out, we've got to break those bonds in a chromosome puff and separate the two strands of the the DNA helix. The DNA unwinds there and then starts its replication process. In order for this to occur, we have to have an enzyme that catalyzes this reaction. The enzyme that catalyzes the reaction, polymerase. Polymer, as you know, is a molecule of repeating units, and the repeating units here are nucleotides, A, T, C, and G. ACE at the end of a word refers to an enzyme, and we're going to be building a molecule with this enzyme. The DNA polymerase joins the individual nucleotides to produce a DNA molecule, and then it actually makes sure that there are no mistakes. Obviously, that's a pretty complicated process we're not going to talk about here, but generally speaking, we say that it proofreads each new strand of DNA to make sure that each new strand is identical to the other. As you get older, this proofreading process gets a little bit strained and we make more mistakes. That's one of the reasons why we believe that older folks generally get cancers more often than young people, because of this difficulty in proofreading as you get older. So that's the basics of DNA replication. Thank you, and adios, amoebas.